Okay, so this lecture will cover translation. So remember, translation is the process of going from our RNA transcript over to our protein, right? So we already talked about what happens when we write down a copy of that gene. That was writing down the recipe from this big old recipe book. But now let's talk about how we're going to actually make a functional protein from just some instructions. So how do you actually bake the cake, bake the pie from the recipe? That's what translation is, the process of synthesizing that specific polypeptide or making the product. So if you saw this and I said I have a map that shows you exactly where an A in Bio 102 is at, you just have to follow the instructions, what would you need to interpret this? I hope you said you would need some type of key, right? Some type of key that lets you know what this symbol, to let you know what this symbol means versus this symbol, right? You need something to kind of help you out. So that's what we're going to be using. We're going to be using a key that's going to help us understand that long code of sequences that we've been making through our mRNA and help us to make a protein from it. So the long code that we've been making is called a genetic code. Genetic code, right? This is AUCCGCAGG, right? That is our genetic code. Very important. This genetic code is read in a particular way. It is read in groups, and those groups are called codons. So these are groups of 3. So every time we see a genetic code, right, I'm going to write one across the top, AUGCCAGAG. Every time we see them, we always want to read them in groups of 3 because that's how you read these codes. They're called codons, groups of 3. So every time we see these codes, we want to automatically separate them into groups of 3 or codons. These codons are going to all specify a very particular amino acid. So each of the letters each of the groups of 3 are codons. So each group of 3 letters will represent some type of amino acid. So if you see on this list here, we have amino acids such as methylene, valine, alanine, glycine. What you see here on the right is the 3-letter code for what they're called. And in the middle, you're going to see the actual codon that makes it up. Some codons are going to tell you when to start making that polypeptide, and some are going to tell you when to stop making the polypeptide. So the start codons are going to be pretty much AUG. A great way to remember that is think about when school starts. School typically starts in August, right? So that's a great way to remember AUG, or the start codon for this entire sequence, AUG. There are multiple stop codons. So you have UAA, you have UAG, and you have UGA. So anytime you see that codon, that means we're stopping our process of translating that mRNA code. And this is what we call a generic code, which means that more than one codon can specify a specific amino acid. So you can see that you have multiple codons that are going to make THR, right? So there's going to be a lot of different ways to get the amino acids that we have. So let me show you how to read this chart, because you definitely will have questions in which you have to translate a particular sequence of DNA, and of RNA. We'll go through that. So the first thing you want to do on this chart, now this is important, is you want to, let's say, I'll just give you a random sequence. I'll give you a few so you can practice on your own. Just gave you 3 random ones. So we'll start with the first one, which is UGG. So the first thing you want to do is, once you have identified the group of 3, right? So when you get a code, and we'll do one, you want to start marking off groups of 3. That's step number 1, mark off groups of 3. Then you want to go through each group and identify what amino acid they make. So UGG, we would first look on the left side. This is telling us the first letter. So these are the possible letters it could be. We first find the first letter, which in this case, it is U. So we're going to start right here. So now our answer has to be somewhere on this row. Then you say, what is the second letter? Second letter is G. So we're on our first row. We go over to second letter across the top, G. So now we know it's in this box. And then we just finish the rest of the code. It's UGG. So we find it. UGG is going to make TRP, which is tryptophan. So you would write TRP. The same thing with AGC. So I'll do that one. First we find, oh, here's A. We know we're going to be in this row. We find G. Our G is here on the end. And then you find C. C is here. That's serine. S-E-R. So those two are going to be serine. I'll save the last one for you. It's CAA. I forgot the line in the middle. So again, make sure you're very familiar with how to read these. So again, let's talk about how to translate our mRNA. We don't have too many slides here, but I want to kind of walk you through that process again. So we're going to start with bacteria. It's just easier to understand, so you get the basic concepts instead of talking about eukaryotes. So all of our mRNA is going to have a ribosomal binding site. So this is where our ribosome will bind. So remember, our ribosome is those little dots we would always see. This is where it's going to bind. It is going to start at the start codon. So this is going to be the first amino acid in the polypeptide sequence that begins with UAG, right? So you may have some codons that come before it, but we're only starting when we see the start sequence. The coding sequence is all the area in the middle. It's just all the other codons in the middle. And then finally, our stop sequence or termination or nonsense codon is going to be the last one that contains these values. So again, there can be some after it, but once we stop it, we're done. So we're only going from start to stop. If there's codons on this side or on this side, we're not worrying about it. So again, this is just showing you a real-life picture, right? And how the start codon is going to define some of the reading frame. So what we want to do here is anytime we see this, we want to make sure that we can, mark off our answer in threes. And then once we do that, we'll identify our first codon and then transcribe the rest of the message. This is interesting because it's showing that one small change, let's say a mutation somewhere, can really mess this up. So if it's a group of 3, and let's say I added a U here, right? On the bottom, there's a U. On the top, there's not a U. That shifts everything down. And because they're read in groups of 3, that can change all of the amino acids. So that's why things like mutations can be very bad for us, because it can cause a totally different protein to be made instead of what was originally supposed to be made. All right, so this is an example here that I want y'all to work out. I'm going to put it up on the screen for a second so that you can pause the video and work through it. I'm going to also try to help you and give you some steps. Steps number 1 is separate the codons. So automatically you want to start going along and separating codons by groups of 3. Second step is to find the start. Third step is to translate them, right? Use the key below to translate them. And the fourth one is to stop. Simple as that. Separate the codons, find the starting codon, translate everything in the middle, and then stop. So I'll give you about 5 seconds and I'll start this problem so you can pause it at this point. All right, so I'm going to start working on it. If you've worked on it, I hope you have. The first thing we want to do again is to separate this message. It's going to be helpful for us so we can really truly identify how everything is being read. This is good because I'm probably going to try to trick you by putting a false AUG. You see this right here? If you didn't separate it, you would have thought we started right there, but we're not. So we're going to separate all the way down. Step 1 is separate all the way down. Okay. Step 2. That's done. Step 2, find the start. So we know start is AUG. It's saying met here, but this is our start. So make sure you always know that start. It has it in green. You kind of forgot, but that's where we start. So you find the AUG. It's not here. It's not here. It's not here. Oh, here we go. So the first protein we're going to translate is met. Right here at the bottom. CAU. C, we find C. We find A, we find U. That's hist. CAG. CAG. GLN. GAU. UGA. Oh, stop. We don't care about the rest because we already stopped. So, these are going to be the amino acids that we made with this particular message. Met, hist, GLN, aspartame. If you got a question that said, how many amino acids does this polypeptide make? 4. What's the third amino acid in this sequence from the polypeptide you made? Glycine. It is not the third one here because we didn't make this one, right? We only started on this part of the actual polypeptide. Make sure you understand that, okay? So follow these steps. If you follow these steps, when you get questions about the polypeptide you made, right, because this is the polypeptide, you won't be confused. Make sure you understand that. Okay, so again, let's talk about the mRNA and rRNA molecules that are involved in this process. So mRNA is going to give you the codon, right? It's going to be that message that I just wrote on the top of the screen before. This is going to give you the set of those 3 nucleotides. So that's the green line here. The tRNA, like I mentioned before, it is going to be the transfer RNA. It's going to be what carries the amino acids. But on the bottom of the tRNA, there is something called an anticodon. This is the 3 RNA nucleotide that is anti-parallel to what we're seeing here. So for instance, AAA from our mRNA is going to have a tRNA that is anti-parallel or opposite of it, forming UUU. So this code for AAA, it should make lysine. Let me go back. So AAA, it will make lysine, LYS. And if you see on this image here, the UUU correlates with a lysine, right? So that's how we're kind of connecting everything together. And then finally, the rRNA, which is essentially your ribosome. This is going to allow the tRNA and the mRNA to come together, and it's going to accept it, and it actually kind of help to form the polypeptide. So think of like a ticket machine, if you remember those. Let's say you want a stream of tickets, you can put your tickets in there, or even, a cash acceptor. If you put $1 in there, it's going to kind of pull in that code right here and allow it to be translated up top. So again, just kind of reinforcing that there's a lot of components we have here. mRNA, tRNA, ribosomes, which will represent the rRNA and some of those transcription factors. This is also a chart from your book, which kind of describes it a little bit better if you want to review that. And then also just kind of quickly highlighting the steps or stages of translation. It has the same ones as transcription, which would begin with initiation. So first the mRNA, tRNA, and the ribosome come together. Elongation, we go from start to stop. And then finally termination, everything separates, and then you have your final polypeptide, which will then start its folding process and then be functional like we've talked about before. Again, kind of showing the overall idea, or at least big picture of what you're seeing. So you begin with your DNA, which is anti-parallel 5' to 3', right? You see the matching or the correlating nucleotides. You transcribe that to make a mRNA molecule, right? We lost our T's. It's going to be more similar, or at least we're going to have our U's instead of our T's. You have your mRNA. You first separate everything, and then you're going to create or find your start codon. You translate your start codon until you get to your stop codon. You do that by using those anticodons, which are anti-parallel to the to the mRNA message here. those tRNAs are going to then have the amino acid attached to them, and you will eventually create a polypeptide that can be used to go through the folding process. So looking at this picture, talking out loud to yourself, seeing if you can walk through each of the stages will be a great practice for you. So as we close translation, I wanted to bring up something that's extremely relevant for us, especially at this day and age, which is the new COVID mRNA vaccine. So as a reminder, I am an immunologist, so I study our immune system. So outside of what we do for Bio 102, I'm pretty well versed with this information. So I wanted to make sure I shared this with you so that you can be more informed citizens. You can share this with family and friends, and you can apply what we've learned in class to a true real life relevant scenario. So conventional vaccines, think of the flu vaccine, work in this manner. We take either a weakened pathogen, so we've taken the flu and we've taken out proteins to make it as strong and as infectious as it normally is. So we weaken the pathogen. We've either inactivated the pathogen, so killed it, or done some other manipulation to it, or we just take a tiny piece of it called an antigen and put that in the vaccine. So either way, all of these are different things that we can use to activate our immune system, to get our immune system to recognize this as being foreign, and then create antibodies or create an immune response so that when we come across the real flu, we already have a lot of soldiers or antibodies in our body that can be used to fight it off. This is why sometimes when you get the flu shot, you feel a little sick because you're actually getting either a dead flu particle or a portion of the flu that will cause you to be sick. This has worked for the flu, but it will not work for something like COVID-19, which is a lot more dangerous. Some people, if they get just a portion of COVID-19 through an antigen or even the weakened pathogen, that can still cause a lot of damage. So we cannot use this normal method of vaccine generation. Instead, we've used an mRNA vaccine. So you just heard me talk about mRNAs a lot. So I want to remind you how this vaccine is different and how it works. So what we've done as scientists is we've identified one of those spike proteins on the SARS-CoV-2 virus. Once we've identified that protein, we actually looked at the message. So remember, we've already just talked about the protein, right? Central dogma, right? We have the protein. Before the protein, you have the RNA. And then before the RNA, of course, you have the DNA. So now that we know what the SARS-CoV-2 protein looks like, we identified what the mRNA transcript or message would look like. So, because we know what the instructions are to make this protein, our mRNA virus that we have now is where we give our own body cells this instruction, this recipe, to make the protein. So the vaccine consists of mRNA, which we can give to our own cells that are packaged through these lipid nanoparticles, which are just little fat particles, so your body won't destroy it before it gets to the cell. Once this mRNA message gets inside of your normal cell will use its ribosomes to make little tiny spike proteins. These spike proteins will then be in your body, and you will recognize them as foreign because you made a protein that you don't normally have, and you will create an antibody response to these spike proteins. That antibody response will allow you to make millions of antibodies towards COVID. So whenever you come across the real COVID-19 or SARS-CoV-2 virus, you will have already seen these spikes because you made them, and you will already have abundance of antibodies in your body to be able to protect you. So this is really bringing together a lot of stuff that we talked about. By taking the mRNA from the vaccine and giving it to your body, this eliminates a lot of the damaging effects that you see here. Using your own body to make a little portion of it will not be as dangerous as taking a real portion of the actual pathogen. mRNA, it's not going to get into your nucleus because remember, all of this translation happens in your cytosol. So you don't have to worry about the mRNA going into your own DNA and incorporating itself. It's all happening in the cytosol. You're just taking advantage of the ribosome in your own cells to make little small similar spikes so that you can create an immune response so that when you come across the virus, you can be more protected. So I just wanted to kind of close on that thought because this is so relevant, so critical that we are informed. And I want you to kind of see how a lot of the stuff that we talked about in class and those concepts are really all around us. We talked about that on day one, how biology is just truly surrounding our life. So I wanted to just give y'all a little quick summary of how that vaccine works. So when you're watching the news, when you're talking to family and friends, you can explain to them how this is less dangerous because you're not using the real virus, how you're actually using your own machinery by taking the instructions for the protein, making that protein yourself, then using that protein to be displayed so that you can create an immune response. All of these things, I just want you all to be informed. So that concludes our lecture for right now. So make sure you review it, you look at everything related to translation, and if you have any questions, save them for class.